According to the Natural Resources Defense Council, important waterways lead to over 117 million Americans. With these statistics, it is important to know how water pollution, which can be found in these waterways, affect our bodies. The purpose of my experiment was to be able to show that the heart rate of Daphnia magna is in fact affected by different concentrations of copper sulfate. I hypothesized that as the concentrations increased, the heart rate would decrease. Daphnia magna are very useful in heart rate research because their heart is easily visible, seen here beating in the back. I split my Daphnia into 5 experimental groups. The control, 10 parts per million, 100 parts per million, 500 parts per million, and 1000 parts per million of copper sulfate. After 48 hours of exposure, I then measured the heartbeat. I then analyzed my data by graphing the heart rate of the Daphnia over the concentration of copper sulfate. The standard deviation was represented by the error bars. This Daphnia shown was exposed to 500 parts per million of copper sulfate. As you can see, little color disfigurations exist along the body. My studies show that Daphnia's heart rate is actually increased as the concentrations increased. This suggests that pollution in our bodies could also affect our heart. With our knowledge of the effects of copper sulfate, we can expand and use new pollutants such as lead and zinc. Additionally, we can use different solutions in conjunction with these different pollutants. We could use our knowledge of how these pollutants affect our bodies to help cure diseases related to these pollutants and save lives around the world.